Yeah, thank you for having me. My name is Zhang. I'm one of the neuropathologists working at the University Hospital at London Health Science Center. I have no conflict of interest to disclose. So, because my talk is on lung cancer brain metastasis, so before I go to my research, I want to give some brief introduction about the general lung cancer and metastasis of the brain. So as you can see, I pulled some figures from the Cancer Society of Canada. So you can see here, as of 2016, cancer is still the leading cause of death in Canadian population. It accounts for about 30% of the total population death, followed by other and cardiac disease. And according to the 2019 Canadian Cancer Society number, so lung cancer is the top one on the list in both male and female. So what is lung cancer? I mean, so everybody is talking about lung cancer every day, so we know more or less something about lung cancer. So lung cancer is actually a very heterogeneous group of tumor. If you ask me, a pathologist, I can give you, there's more than 20 different types of lung cancer. Just to simplify it, and for treatment purpose, we group them to small cell carcinoma and non-small cell carcinoma, because the treatment is very different. So for my research, I only focus on the non-small cell carcinoma part of the story, okay? So the major risk factors, as we all know, is tobacco, is smoking, and also occupational exposure to asbestos and all those, and some genetic background as well. So I won't go to the detail of those. So lung cancer, if you identify them early, if you tackle them early, they won't kill you, right? If you found them in a small nodule in the lung, you can resect this part of the lung. You can resect maybe a lobule of the lung, even the entire lung on one side. The patient can still survive. The main reason for lung cancer is so lethal is because lung cancer, they want to go to other side of the body. This is the process called metastasis. So the one preferred location is actually the brain. So lung cancer brain metastasis is one of the major complications of lung cancer, actually can cause patient demise, okay? And other side, like liver or bone, and can be also resected, but once the tumor goes to the brain, it's hard to treat. So metastatic brain tumor, again, is a heterogeneous group. Any systemic malignancy goes to brain is considered metastatic brain tumor. The presentation is different, depending on the location of the tumor in the brain, if it's frontal lobe or brainstem or cerebellum, so presentation will be very different. Could be like nausea, vomiting, could be very non-specific, like numbness or tingling, right? That could be weakness, and fatigue even, and memory loss. So very general, very non-specific presentation. Having said that though, it doesn't matter how subtle the presentation is, once you have brain metastasis, it's considered stage 4 disease, meaning it's a very late stage, and bear a very poor prognosis, and the treatment usually aiming for palliative, not for cure. So how does a systemic tumor going from like lung or skin or elsewhere in the body go to the brain? So they have to follow sequential steps. So number one is they have to actually proliferate on the primary site, and they have to break through the so-called basement membrane. So this is a material to confine the local growth. So once the tumor breach the basement membrane on the local site, they have to go to the blood vessel. So once they're in the blood vessel, they have to really survive the your immune surveillance, because we have so many immune cells, like lymphocytes, neutrophils, and natural killer cells. They are there to target the foreign material. So for tumors, they have to really survive the immune surveillance, and they form a tumor emboli, and again attach the blood vessel wall in the distant side, there is their second home, such as the brain in this case. So once they reach the second home, the brain, and they have to go through the blood vessel again and enter the brain and survive. So brain itself is not a very friendly environment compared to other sites because it is so different. The microenvironment is very different in the brain compared to the lung and other areas. So there's a lot of different layers of defense mechanisms in the brain. So I just listed this. I borrowed this picture from a review article. So there's macrophages, and there's dendritic cells, neutrophils, lymphocytes, and astrocytes. The traditional view of astrocytes, because we have a lot of very quite abundant astrocytes in the brain. So the traditional view is they actually become reactive and to protect the brain from the metastatic tumor cells. But very recently, people actually found the metastatic tumor cells, such as the lung cancer, once they're in the brain, they actually become very sneaky. They form this sort of gap junction. They form a junction with astrocyte. So the astrocyte, instead of prevent them, they're starting to provide nutrients. They're starting to give them nutrients and glucose and to start to facilitate brain mass. So this research, I was just focusing on the gap junctions between the metastatic cells and the local microenvironment. So just a very brief one slide on gap junction and related proteins. We focus on connexin and pannexin. Is a group of different proteins, and they share very similar molecular structures. They all have 4 transmembrane hemichannels, like this is pannexin, connexin. And connexin can form a gap junction, and pannexin actually just become a hemichannel. They don't form with other partners. So we pick 2 molecules, and connexin 43 and pannexin 1. The reason for that, they all have relatively higher level of expression in the astrocyte of the brain. So the hypothesis is, if the lung cancer in this case, they want to survive in the brain, they have to form some sort of junction and to adapt the microenvironment. So we would think the connexin 43 or pannexin 1 will be upregulated, therefore to facilitate this process. So we were going to examine the brain metastasis, compare with the primary tumor site of the lung, and also compare with if the patient has lymph node mass, we also compare with that, and also compare normal brain tissue with the microenvironment of the tumor. So to do that, we divided this to 3 different steps. So as I explained, the first step will be characterize the expression level of pannexin and connexin in these different locations, and then we will analyze the different tissue types, and also in the end correlate with the patient survival. So let's start with a patient cohort. So as a pathologist, we have a big collection of archive, which are the patient tissue get resected, and we keep them in the pathology department as an archive. So we search the past 18 years of the pathology department for any brain metastasis tumor with lung primary. So this is the first we generate a list. So among this list, I will look for whether or not the primary lung cancer was diagnosed on site, meaning we can have actually the same patient, but with the primary lung cancer also available, so we can compare. Because a lot of patients, they actually diagnose elsewhere, so we won't have the tissue available. So once we have a paired group, primary lung cancer and metastatic tumor, so I will review all the slices and to exclude all the tumors with abundant necrosis, which is not workable, and we will include all the with viable tissues, so we can extract protein, we can do other tests. So as you can see, this is a table of our cohort. We, in the end, in the last 18 years, we found 61 pair of cases which are actually workable for our purpose. So there's a slight female predominance, about 60% of them are actually female patients. And the age, they ranging from 46 years old to 84. to 82 actually. Histology type, most of them are adenocarcinoma and squamous cell carcinoma is also there. 17 patient has positive lymph nodes. So what interests us is actually there's a time interval. This time interval is calculated with the primary lung cancer diagnosis as a start point, and the brain mass development as an endpoint. So actually, the time interval is a big range. And some patients present with concurrent lung cancer and brain mass. Some patients, they have their lung cancer diagnosed like 10 years ago and developed brain mass like after 10 years. So it's quite a long time interval. And in terms of brain metastasis location, it could be anywhere in the brain, like cerebellum, brainstem, leptomeninges, and so on. So this pie chart is just showing you some of the tumors have molecular workup, meaning we will have the status like EGFR and ALK mutation. Those are the so-called personalized medicine, right? So we treat with a specific target. But only a small population of the case has this available because most of the cases are actually dated back to 18 years. The tests are not available yet. So I'll just give you one example of one of the patients so you will understand how we take the samples. So this is patient GC, is a 60-year-old female, initially presented in the 2014 January as shortness of breath and hoarseness of voice. So they went to the family doctor and did a CT scan, and they found this nodule on the right upper lobe of the lung. So they did biopsy and turned out to be adenocarcinoma. So the thoracic surgeon took this out and pathology department received the specimen, and so we did a thorough examination. Again, confirmed adenocarcinoma of the lung. So a patient received chemotherapy, and about 1.5 years after, that was in 2015, November, the patient had actually had a brain aneurysm and was trying to coil the aneurysm. So they have MRI scan, and they found this, a ring enhancement of the brain tumor in the cerebellum, and this is also resected, turned out to be adenocarcinoma of the lung, and it's a metastatic tumor consistent with lung primary. So the patient had a resection and radiation afterward, and then about 6 months after, 2016, and she had another MRI scan, and there's a second focus developed on the opposite side of the brain. This time is on the frontal lobe of the brain. So again, this was resected and turned out to be the same tumor. So 6 months after, it developed on the other side. So well, unfortunately, in 2017 September, she had another scan. There's another focus developed. So this is on the parietal occipital lobe. So luckily, this time is not a metastatic tumor. This instead is we call it radiation necrosis. So it's more like a complication of the treatment. The patient is still alive at this moment and just having some seizures. So I'm just giving you some example of how data look like. So this, it didn't project very well, but so this is the lung adenocarcinoma resected back to 2014. So this is the cancer. You can see there's very, well, it's details. I don't think you can you can tell. This is epithelial tumor and forming this like large sheaths and nests. The normal lung tissue is not there anymore, being replaced by this solid tumor. And at the same time, there's a lymph node. You can see this is the lymphoid follicle, and there's also cancer there. So this was 2014, and back. So if we look at the case of 2016, there's a brain resection. And I draw a line here to show you this is where the metastatic tumor located, and this is where the brain parenchyma is. So this is actually the microenvironment providing all the nutrients, everything to let this tumor grow. And if you look at high power, the morphology looks very similar as the primary cancer. Okay, so this is how we pick our tissue to make sure they actually viable and we can get something out of this. So I use this to explain how did we study this. So we built something called a tissue microarray. Because we have so many different sites and so many patients, so we want to make our life a little bit easier. So we examine them under the microscope. We pick up the area of interest. So we take a little tissue core from each of the tissue blocks. So we put them all together. So we line them up. So this one line, one row is one patient. So we pick 3 cores from each block. So we have a replicates of 3 in this represent lung adenocarcinoma, 3 represent lymph node, and 3 represent the brain metastasis, and the 3 core represent the so-called microenvironment. So this is how it looks like once the product is done. So on this one slide, we have built-in control. We also have like 10 patients. So 10 patients, each of them have primary site, lymph node mets, brain mets, and also the microenvironment. So we can study them all at the same time in a very consistent way. So it's a fairly high throughput analyze method. So we use different antibodies to target different antigens, and we use to explain a little bit, immunohistochemistry, we use antibody, primary antibody, secondary antibody, and the color will be different. The brown color is positive, meaning there's a protein expression there. And we use the software to analyze how much protein is expressed, and depends on the intensity of the expression, how many cells are actually positive for this antibody, and so on. It's a relatively complex process. I won't bore you with that. I just show you one core, and this is that core. So this is how it looks like under the software. So this part is the positive, and this part is the negative. So actually, before we jump to the panexin and connexin expression pattern, we want to make sure this tumor in the brain actually reflects is the same tumor in the lung. So we want to see if the biological characteristics change over the 10 years. So we threw in some markers like CK7, CK20, and TTF1. So the classic lung cancers, they are CK7 positive, 20 negative, and TTF1 positive. So this is the lung adenocarcinoma. You can see in the same patient in the brain, their biological characteristics, they are still there, is maintained. So they show the lung adenocarcinoma feature. So now we can talk about the panexin expression. Again, the tissue core, and this is the immunohistochemistry on the bottom. And there's actually a significant increase in the panexin-1 of the brain metastasis if you compare with the primary lung cancer. So meaning that panexin-1 is upregulated. What surprises us is actually there's a lymph node metastasis also have this similar level of panexin-1 expression. So meaning this is not specific to the brain. So next we want to know there's any difference between the metastatic tumor bulk and the microenvironment. So there's no difference, as we can see from here. And then the next step we check is this higher level of panexin expression unique to this microenvironment, or also upregulated in the other side of the brain. So we compare the normal gray matter and the white matter, and that's you only see them in the tumor microenvironment, not in the adjacent brain, normal brain parenchyma. So connexin, however, is we would like to see a significant difference, but on the other hand, it's actually not so much upregulated. So meaning panexin maybe play a major role in this gap junction formation. So since we know the panexin expression level has a different big range, and also we know the patient, the time interval is so different. So we wonder, can we use panexin as a predictor for how aggressive the tumor is? So we draw this survival curve. So we use the start point as the primary lung cancer diagnosis, and we use the end point as the patient develop brain metastasis. There's actually no real correlation between the panexin-1 expression and this time interval. So just to summarize, there's no difference in connexin 43, but there is a significant increase in the connexin 1. However, there's no correlation with the patient survival. So in the future, we just did a very preliminary study on the expression intensity. But as we know, connexin and pannexin, they are supposed to express on the cell membrane to form gap junction. But in some study, they say in the actual brain, in the microenvironment, these two proteins can actually be mislocated into the cytoplasm or in the nucleus instead of on the cell membrane. So we want to look into the cellular structures. Where are they located? And also we want to compare these two proteins in other brain metastases. In addition to lung cancer, we want to look at breast cancer, we want to look at melanoma, kidney cancer, and so on. So yeah, this is a short summary of what we have done so far, and we want to thank Brain Tumor Foundation for support this work, and also this was partially supported by Department of Pathology as well. And we want to thank Dr. Gail Lard and Sylvia to providing some antibodies and consultation, Linda Jackson for technical support. This is my lab, and this work is preliminary done by Somia, who is an undergraduate student in our lab. This is this is her thesis, fourth year thesis work. Thank you. I would like to